like everyone else, I've crammed 30 minutes into 10 and I'm going to zip along. But it's important to kind of give a unique perspective at LUMCON. I'm right here. I'm living in the middle of the mobile mud, as Bob would probably say. And this is a great photo that I'll get back to. This is on the way to LUMCON. Those of you that have been there will probably understand it. Come on, go. It's not moving forward or backward.
Sometimes I reset it.
Okay, so very simple. I'm going to start at the fringe, and then we're going to work our way out to the coast. So when you start wondering what I'm doing at the fringe, this is why. We're going to work our way out, and it's going to make sense, I promise. And then we'll get to some conclusions. For those of you who haven't seen LUMCON, it looks like an airport terminal in the middle of nowhere, in the middle of a marsh. But it's an awesome place. It's got a lot of cool geochemical and microbiological toys, small vessels. We have the Pelican as well. We're building a regional class research vessel. So a lot of cool things happening in the middle of nowhere. But it makes a great site because I can walk out of my office in boots and just take some samples and have a lot of fun. So what we're taught this is mainly for the international folks here that might not know this area quite as well. So we're actually taught that Louisiana looks like a boot, but this is actually what it looks like in much of this region. So it's kind of missing its toes, I would say. This is LUMCON right here, where that little arrow is. Can you see any land? So it's missing its toes, right? And it's all mobile. Again, this is what you see when you survey the area from a plane. And I did want to get my pilot's license until we got hit by a frigate bird on one of these flights. That was very scary. But anyways, so with this all in mind, and me being a methane head, I like all things methane. I'm going to transition you into the marsh. But first, we're going to talk about some of the changes that we're already seeing over here at the coast. This is a USGS elevation marker. This is at LUMCON. It's been there for 40 years now, since the building opened. Subsidence and also land loss in general is really massive and terrible in Parish. You can see saltwater intrusion here. You can see these old trees getting the water sucked out of them by the salt water around them. That's a common feature in the marsh. I showed you guys this at the opening. 40 years ago, it was a cow pasture. Does that give you, it's open water now. That's what we're working with here. This is me in 2017 with my 3-month pregnant wife, giving her a piggyback ride into the building because we have nuisance flooding a lot that floods our parking lot. And this is a time series look at that over the past couple of decades. So that's what we're working with. A lot of mud moving around, a lot of water. And now we'll get into some science. So at the fringe, salt marshes, this is what you see. This is Spartina. There's some juncus hanging around here as well. But in one of our NSF funded projects with folks from University of Tennessee, LSU, and ODU, we record about 15 sites expanding from LUMCON all the way to the eastern bay over toward Port Fourchon, for those of you that are familiar with it. What we noticed, no surprise, was a lot of methane approaches saturation at depth. Sulfate reduction or sulfate consuming processes probably occurring a bit, but sulfate is running out at depth. So we had that in mind, but again, we wanted to go out to the coastal ocean. And with all this mud moving around, what does this mean for the methane cycle was one of our big questions. And we chose to approach it like classic biogeochemistry, looking at the cycles that we normally predict in a sequence that you would see here. And we know that there's methane in this water. My PhD advisor actually published this paper a few years ago showing 400 nanomolar concentrations of methane in the water column in the summer. You can see it actually in the fall, it actually gets much lower. But to give you some perspective, atmospheric equilibrium is about 2 nanomolar. So this is actually a lot of methane that's dissolved in this water. So we were interested in looking at the biogeochemical processes. We know in these sediments that very often they run out of oxygen in the first couple millimeters, the first few millimeters, a lot of organic carbon input. Of course, iron reduction is very important in this area, but also sulfate reduction. And then we go into a zone called the sulfate methane transition zone. That's where methane is being oxidized anaerobically. And below that, you have methanogenic zone. That's the classical look at biogeochemistry. And our approach to looking at this was to study 5 sites along a transect with a varying amount of sedimentation rate, so sediment inputs. And this was from a modeled paper, Matthias Eggers, over 1,000 cores. It's about the best representation we have. It includes satellite imagery of POC. It also includes depth as an empirical relationship to solve the sedimentation rate. You can also look at the sulfate methane transition zone depth with a similar algorithm from Egger. So we chose these 5 sites, went out on the Pelican, May 23. We're going out again this year. We had Dr. Mighty's profiler and lander. He was working with us on this. We also collected multi-cores and gravity cores. I'm going to gloss over a lot of the geochemistry. We can talk about those methods later, but some of the more specialized things that I'll mention, we do have or have started doing some reactive transport modeling on these cores so we can understand these oxidation and reduction events and the seasonality that's occurring in the sediments. But we also have radio tracer rates of methanotrophy, so that's methane oxidation. These are the most sensitive assays you can actually do, and sulfate reduction. And what we saw when we went in May 23, this is the water column. Yes, there is a lot of methane. We're popping up at 100 nanomolar in May. Sites, they're MEF 1 through 5. MEF 1 is closest to the river. 5 is furthest away. Coastal ocean sediments. So when we started looking at the sediments, what we saw was very little methane at all in May, like one micromolar, which is tiny. The sulfate profiles showed no actual depletion or removal. Again, you got to think reoxidation. You have really intensely coupled iron and sulfate cycles, sulfur cycles in this upper layer. And we know this because we actually measure the rate with radio tracers. So you actually see we're up to 20 nanomole per cubic centimeter per day, which is not a slacker rate. It's a pretty robust rate. So it's happening, but it's just getting reoxidized so fast in there that you never see a reduction in the profile. Gravity cores, you can actually see a little bit of depletion, but you never see methane above 2 nanomolar in May in this particular study. So we're drilling down on this question a little bit more. We got my postdoc is here today, Dr. Chiquita Brooks. You can ask her about this. This gives you a little preview of what she's into right now. We're looking at MCRA copies. This is a functional gene that's responsible for methanogenesis or methanotrophy. It can kind of go either way. We have the benefit of actually having these radio tracer rates, so we can again further distinguish that, hopefully. And you'll see the gene copies go up to 10 to the 5th, which is also not a slacker number. And it's corresponding with where we actually see no methane oxidation. So this is a unique situation where you have non-competitive methanogenesis occurring in an iron-rich sediment. And it's we're actually drilling more into that with the seasonal work, which is made possible again, Chiquita's, Dr. Brooks' work. And we got to thank Jeff Krause and all the qualified PIs on this. You guys have made it possible for us to do some really cool seasonal work at one of our sites. This is one of our sites, and we've been there 4 times now. And this is sediment data again. And we're seeing that this is a beautiful time series, actually. If you look at the methane, July 24, we're peaking at 10 micromolar, most we've ever saw. It's like party time. And then, you know, as you go backwards in time, so seasonally go backwards, or I guess you could say backwards, forwards, whatever. You know, you've got a little bit less in September, but January and May are quite similar. Interestingly enough, the temperature, bottom water temperature at both of those times was 22 degrees Celsius. Bottom water 22 degrees, and is that all? This is the fastest I've ever taught. But we can see this really awesome iron peak too that kind of coincides. So again, we're starting reactive transport modeling on this, and we're seeing some good rates. We had to jack up the bioirrigation coefficient to actually get this to move. And this is work from Christoph Malley and Maruf, his graduate student at UGA. So conclusions are, we have some cool non-competitive substrate stuff. This might be fueling methane in the water column. And global models led us astray because we didn't find an SMTZ.